having a great day. We are Group 5. And for today's vlog, we are going to talk about gene therapy. Let's get started.
What is gene therapy? Gene therapy is a medical approach that involves manipulating or modifying genes within a person's cells to treat or prevent diseases. It aims to introduce healthy genes or correct faulty genes to restore normal functioning in the body. Cell therapy, on the other hand, involves the use of cells, typically from a patient or a donor, to treat various medical conditions. These cells can be modified or manipulated before being administered to the patient, and they can help repair damaged tissues, boost the immune systems, or provide therapeutic effects in the body.
How does gene therapy works? This new genetic material, such as working gene, is delivered into cell using a vector. A vector is like a package used to deliver a specific message. Viruses can be used as vectors because they have evolved to be very good at getting into cells. Scientists have learned how to remove the viral genes and use the same ability to treat or prevent disease. In this case, their goal is to insert the new genetic material into the cell. The vector can be delivered in one of two ways. Ex vivo treatment removes a person's own cells and delivers the genetic material to these cells outside the body. The modified cells are then returned to the body. In vivo treatment means the genetic material is delivered directly into the person, such as through an injection. If you will ask if a viral vector can make you sick, the answer is no, it can't, because before a virus can be used to carry therapeutic genes into human cells, it is modified to remove its ability to cause infectious disease. All viral vectors are tested many times to safety prior to being used in humans.
Current strategies for gene therapy are mainly based on the following. Number 1 is immune regulation. The gene of antibody, antigen, or cytokine is introduced into the patient's body to change the immune state and achieve the purpose of prevention and treatment of disease. Number 2 is gene silencing. Using antisense RNA, ribozyme, or nuclear phthalate to inhibit the expression of some oncogenes, inhibit the proliferation of tumor cells, and induce the differentiation of tumor cells. Number 3 is gene replacement supplemental. The target gene is transferred into the diseased cell or other cells, and the expressed products can strengthen or correct the function of the defective cells. Number 4 is gene replacement complete. The normal gene is used to replace the pathogenic gene in situ, so that the DNA in the cell is completely normal and restored. Number 5 is gene repair. Directly repair the abnormal part of the gene.
Gene therapy is a technique that modifies a person's gene to treat or cure diseases. Gene therapy can work by several mechanisms, replacing a disease-causing gene with a healthy copy of the gene, inactivating a disease-causing gene that can also functioning properly, introducing a new or modified gene into the body to help treat a disease. Deliver gene that speed up the destruction of cancer cell. Supply gene that cause cancer cells to revert back to normal. Deliver bacterial or viral genes as form of vaccination. Provide genes that promote or impede the growth of new tissues, and deliver genes that simulate the healing of damaged tissue. Gene therapy products are being studied to treat diseases including cancer, genetic disease, and infectious disease.
These are the common types of cell therapy. 4 types of pluripotent stem cells and 4 types of multipotent stem cells obtained from adult tissue are listed. Embryonic stem cells, induced pluripotent stem cells, nuclear transfer embryonic stem cells, parthenogenetic embryonic stem cells, hematopoietic stem cells, mesenchymal stem cells, neural stem cells, ileal stem cells, immune cell therapy. How are viruses used in gene therapy? Vectors are typically derived from viruses because viruses have proven to be very efficient at finding their way into cells. In order to make vectors safe to use, all of the viral genes are removed, and vector is modified to deliver on only therapeutic genes. The approach to gene therapy is very different when it comes to oncology and cancer treatment. One way to treat cancer is to provide dividing immune cells with new genetic instructions that can turn them into cells that recognize and kill cancer cells. Another approach is to use vector that can only multiply rapidly in tumor cells. From there, the vectors with specific instruction to kill those cancer cells are available to do so. Thank you.